We, Nitya, Samina, Pushmi, Nitin, have done a project entitled Program Cell Death in Bacillus subtilis in Bacillus cereus species. We have studied two bacterial strains such as Bacillus subtilis N cereus, which are rod-shaped gram-positive endospore-forming soil bacteria. These mesophilic bacterial strains form stress-resistant endospores in order to survive under the harsh conditions. Programmed cell death was observed in these strains under nutrient-limiting conditions, which triggers spore formation. SPOOA, which is sporulating factor A, a regulatory protein which regulates two additional operons, namely SKF, which is sporulation killing factor, and SDP, sporulation delaying protein, governs the spore formation. The SKF operon is involved in the production of extracellular killing factor during sporulation. This operon also acts antagonistically through its products SKFE and SKFF, which confer resistance to killing factor by forming an export pump that pumps the killing factor out of the cells. Inoculation of bacterial strains was carried out in LV media and LV broth fortified with 100 millimolar concentration of each glycine and alanine. It was then incubated for 24 hours on a rotary shaker at 150 rpm at ambient temperature 26 to 32 degrees centigrade. The culture was then grown for 24 hours, was again incubated for another 72 hours under static conditions at the ambient temperature in order to observe rapid cell death. For viable cell counts, aliquots of the culture broth were withdrawn and serially diluted using sterile saline of 0.85% and transferred to LV agar using spread plate technique. Plates were incubated at 30 to 32 degrees centigrade. Then viable cell counts were obtained at the end of incubation period by counting colonies. We have used certain techniques like streaking, serial dilution, spread plate technique, and colony counting. We have also used some bioinformatical tools and databases such as NCBI, which is National Center for Biotechnological Information, Multiple Sequence Alignment, PubMed, and a tool named Classical W. From cell viability studies, we have observed viable cell count reduction from 8 log 10 per ml to 7 log 10 per ml, which is 100-fold reduction at the end of incubation of 96 hours. When programmed cell death inducers such as glycine and alanine of 100 millimolar concentration each were introduced into the LV medium, the viable cell count at the end of incubation of 72 hours from the onset of stationary phase was found to be reduced to around 3 to 4 log 10 per ml. When programmed cell death inducers such as alanine of 0.1 molar concentration was added to LV medium, the caspase activity has been increased in Bacillus cereus from 0.07 to 0.165 and in Bacillus subtilis from 0.06 to 0.154. We have retrieved protein sequences of Bacillus cereus and Xanthomonas species using databases such as NCBI and PubMed, and multiple sequence alignment was performed using Classical W2. We have found the cleavage sites in catalytic domain of cereus and Xanthomonas. The cell death observed in Bacillus species is its stationary phase, where certain bacterial population was eliminated at regular intervals. The interesting fact which we have observed is that the bacteria do exhibit certain molecular features in the stationary phase that are akin to eukaryotic programmed cell death. Further experiments such as Western blotting, protein expression and purification, caspase domain characterization, and signal transduction studies need to be carried out to provide a conclusive proof of bacterial programmed cell death in Bacillus subtilis and cereus species. Thank you.